Good morning, and it's beautiful having you here again. Today we'll continue our discourse. This morning we are talking about fluorescent microscopy. Fluorescent microscopy. I know we've been discussing some of the techniques that we use in microbiological laboratory, in order to do our investigations and experiments. One of such techniques is the use of fluorescence microscopy. What fluorescence microscopy is a method of examining, living cells, tissues, and all these kind of organs using the principle of fluorescence. So the idea behind the use of fluorescence microscopy is that we have dyes that we call fluorescence dyes, and when we stain particular cells or tissues with these dyes, then we mount those cells and tissues on the microscope and view them using different kinds of light sources, like you can use our laser, you can use LED. Now, when we view them, we realize that there's a concept in which particular components of the cell have the ability to absorb light energy and then to emit this light energy in the form of a glow. So, just the way you have like bioluminescence that occurs in some insects, where you see that at night these insects glow. They glow because they've absorbed energy and they're giving out that energy. That's the same way different components of cells are able to absorb light energy and then emit this same light energy, and we see this light energy in the form of glowing or fluorescence, and we're able to view that in on the microscope. And this is really important for us to know. We see that there are different types of dyes, fluorescence dyes. There's rhodamine and there is DAPI, DAPI. These are examples of dyes that we use to stain all these are different cells and structures. And it's important to know that there's what we call fluorophores. These fluorophores are those components that are present within the cell that have the capacity to absorb light energy. Not only do they have the capacity to absorb this light energy, they are also able to give out or give off this energy in the form of a glow. And this is quite important and interesting for us to know. There are different types of microscopes that can be used for these technique. You have the fluorescence microscope, you have the scanning focus microscope, the confocal microscope. All these microscopes can be used and modified in one way or the other to make sure that you can use them to do fluorescence microscopy. So the question will be, why do we want to do fluorescence microscopy? Why do we want to have a situation where you have cells glowing in the dark? Sometimes it's good, or sometimes we use it because we want to look at the shape and form of organelles. We have different, we know that organelles are components of cells, especially our eukaryotic cells. You have membrane-bound organelles. So if we want to look at the shape, the size, the structure of these organelles, sometimes it might be difficult to see it more clearly, or to view it, understand what it looks like more clearly if we are using a simple light microscope or using the dark field or bright field microscope. So our fluorescence microscopy gives us a technique that helps us to be able to view these organelles really well, look at their shape, look at their function. Sometimes we are able to look at some of the things that these organelles are able to do. Sometimes too, when we are studying immunology and want to look at antigen antibody reactions, want to look at how this coupling occurs, in order to form the antigen antibody complexes, how this coupling occurs, what the complexes look like. We can use our fluorescence microscopy to be able to check and study all these kind of phenomena. It's also important in the use of when we study drug discovery. When we look at the mechanisms through which drugs are able to function in living cells, we want to look at the exact component of the living cell that the drug is able to attach itself to. So in doing all these kind of studies, in-depth, serious scientific studies, we can make use of our fluorescence microscopy to be able to view all these cells. Now, these are some of the advantages of using it, but it also has its own disadvantages. It can cause what we call photobleaching of cells. It can make cells begin to lose. Then those fluorophores that are present in the cell, when you keep hitting them with light energy over a long period of time, they begin to lose their ability, they begin to lose their strength to be able to glow in the dark. So it's useful, and it's good that as a scientist we understand what it looks like, understand how to use it and I hope you've learned one or two things. Thank you for listening to this. You have a beautiful day. Remember to like, subscribe, share. Have a blessed day.